Hi, good day. Thank you for attending my webinar, The Innovative Biomarker Spatial Analysis for Comprehensive Tissue Immunophenotyping. My name is Bonnie Phillips, and I am the Senior Manager of the Global Field Application Scientist team at Ultivue. So just a bit on my background, I began at Ultivue in June of 2018, right around the release of our first commercial product. And since then, I began as a field application scientist, and I'm now leading the team. Before Ultivue, both during my graduate school at Tulane University and at UNC, I did a significant amount of IHC-based assay development and troubleshooting, so I understand all of the pain points of these technologies. So thank you for your attention today, and without further ado, I will jump right into my presentation. So, as we're all very well aware, the tumor microenvironment, as well as many other tissue-specific diseases, has a complicated cellular makeup that is not just limited to the complex phenotypes alone, but also their spatial relationship to other tissues or other cells within that tissue. So, specifically in regard to tumors, there are many different phenotypes that make up the tumor immune landscape. And in addition to the tumor cells themselves, there are also immune cells of hematopoietic origin, as well as stromal cells of non-hematopoietic origin. So the immune cells can be divided up into lymphocytes, such as T and B cells and K cells, as well as myeloid populations like macrophages, myeloid-derived dendritic cells, myeloid-derived suppressor cells and dendritic cells. And then there's the stromal compartment, which is made up of cancer-associated fibroblasts and endothelial cells. So not only is the complex cellular composition important in how these tumors function, but also how these cells interact with one another and their spatial relationships can also play a huge role in how cancers progress and respond to treatment. So just to give one example of how important these spatial relationships can be, here I have some data generated by a group at Roche that was presented at SITC back in 2020. So in this study, patients were treated with a novel drug that is a CD137, also known as 4-1BB agonist, while also targeting fibroblast activation protein, or FAP. So FAP is highly expressed on cancer-associated fibroblasts and solid tumors. The combination of 4-1BB stimulation with FAP cross-linking is able to increase function and activation of effector cell populations within the tumor microenvironment. So in patients treated with this therapeutic, it was shown that in biopsies that were taken at both pre and post-treatment from these solid tumors, that in patients that responded to treatment, there were cytotoxic cells that were located within the tumor compartment in close proximity to tumor cells. Whereas in the non-responders, these cytotoxic cells were primarily located in a separate environment from these tumor nests. And this just really shows how the spatial location of certain cell populations is really important. So there are really three main pillars of technology in being able to understand the spatial cellular composition of tissue biopsies. And we can start off with the H&E. So the H&E is still heavily used by pathologists and is a principal method of diagnosis. So it can be used to determine tissue architecture and cell morphology. Although there are shortfalls in that this method lacks the ability to detect protein. So simple immunophenotyping and protein expression, unfortunately, cannot be detected utilizing this method alone. Of course, that's where traditional IHC comes into play, specifically DABs. And these do allow for single, or in some cases, a few proteins being detected at one point in time on one tissue section. So the traditional IHC can be used to identify expression of proteins, as well as simple cell phenotypes. And there are many predictive biomarkers in clinical assays that can be used for diagnostic purposes, such as with PD-L1. Of course, the downside here is that there is a limitation to the number of biomarkers that can be included on a single slide. And this really restricts the complex phenotyping that can be performed, as well as the detection of multiple cell types on a single tissue section. So, of course, this is where multiplex immunofluorescence comes into play. And there are a few technologies that fall within this category. Of course, Ultivue's InSituPlex technology is one of these. So with multiplex immunofluorescence, these allow for the detection of multiple markers on a single tissue section. And this allows for multiple complex phenotypes to be detected on a single slide. So some of these technologies can also show differential expression. And with these technologies, the ability to gain a greater understanding of spatial relationships between different cells in a spatial environment is a feasible idea. So within a couple slides, I will give some real-world examples of how this spatial information is important and has been shown in publications. I am highlighting instances where the InSituPlex technology was utilized today. So I think just giving a brief introduction on how this technology works will be beneficial. So the InSituPlex workflow overview starts off with a single round of antigen retrieval. From there, this is where the ISP technology really begins, because here we start with primary antibodies that have all been conjugated to unique single-stranded DNA barcodes. At this point in time, after the one round of antigen retrieval, we have a single staining step where we add all antibodies to the tissue section at one point. And this can be for 4 antibodies or 8 antibodies or even 12 antibodies. After this single staining step, we then go on to the amplification step, where we are able to amplify the signal. And here, all we're doing is we're increasing the number of tandem repeats. And this process occurs in a controlled fashion, allowing for us to have a semi-quantitative assay. Additionally, it is important to note that because we're working with these DNA barcodes, we don't have to work with secondary antibodies. So there's no need to be concerned about the species in which the primary antibody was developed in, and there's no secondary associated background. And this really allows for us to have a high signal, low background ratio. So once all of the barcodes have been amplified, we then move on to the single detection step. And here, what occurs is the addition of fluorescent probes to the primary antibodies and their elongated DNA barcodes. And all that's occurring here is the set of fluorescent probes hybridizes right to the appropriate DNA barcode attached to the appropriate primary antibody. So this assay can easily be done in a single workday for a 4-plex. And if we were to jump to, say, an 8-plex, it would essentially just be the dehybridization and removal of this first set of fluorescent probes and the addition of the next set of fluorescent probes, which generally takes all of 2 hours. Something else about this technology that I'll be touching on a little bit later is that we're also able to perform a same slide H&E, where all we do here is remove the cover slip and then perform a standard H&E, and then we're able to overlay the images on top of one another, allowing for the ability to visualize the tissue architecture as well as cell morphology of the H&E on the same slide where you get that fluorescent data. So technology highlights is that we have standardized this assay, so it is reproducible, plug and play. The technology is streamlined and simple, easy to use. And the protocol is the same from one assay to the next, regardless of the tissue indication. Sample is fully preserved, as I had shown in the previous slide. There's one round of antigen retrieval at the very beginning. So this preserves the quality of the tissue. It's compatible with conventional pathology, as I had mentioned. Same slide H&Es can be performed. And it is a high throughput assay. So It can be performed on an auto stainer, and many slides can be stained at one point in time. And then, of course, it is scalable, so it can easily be outsourced because it's a very simple, elegant assay that's easy to transfer to different areas. So with that, I just want to start off with this first paper that really shows that with the multiplex immunofluorescent assay, it really allows for a greater amount of spatial information because we're getting all of this data on a single tissue section. So here we have an example of the greater insights that can be gleaned from multiplex immunofluorescence. And here the authors had actually compared the assay directly to a standard IHC. In this case, we're talking about DABs. So in this paper, we're looking at a PD-L1 assay, 4-plex. So this contained antibodies for PD-L1, CD8, CD68, as well as pancytokeratin SOX10 that were all stained on the same slide. And in this paper, there were 50 resected non-small cell lung cancer tissue sections, 50 non-small cell lung cancer patients that were stained with both single as well as multiplex image analysis. So here we then quantified cell density of PD-L1 expression on cells, as well as the consistency of the tumor proportion score. And this was compared by these values that were generated by pathologists. So first, to validate the multiplex immunofluorescence assay in comparison to chromogenic IHC, serial sections were stained. First, there was an H&E, and this was followed by 2 sets of singleplex chromogenic slides, flanked by 1 multiplex slide. So this places the multiplex slide in the center, allowing for a comparison of all single marker slides, with a similar tissue section being at a maximum 1 serial section removed. So next, there was a Pearson correlation test that was performed to calculate the consistency of marker density. And from this data, it was concluded that there was a high correlation in marker densities between the IHC singleplex assays and the multiplex immunofluorescence slide. So that's good. We're seeing concordance between the DABs in comparison to the multiplex immunofluorescence assay. So next, in this experiment, multiplex immunofluorescence really showed a much clearer view of the phenotypic complexity and cellular arrangement within a single section of non-small cell lung cancer tissue section. So the authors really talked about how with the chromogenic IHC technology, each of these markers was essentially in a separate environment where you could not perform a direct marker-to-marker comparison. So now that we had all of these markers on a single tissue section, not only did we get a much higher level of phenotypic complexity, but we're also able to get that spatial information where we're able to compare these markers across the same tissue section. So there, the authors were able to show that, say, PD-L1 expression can now be differentiated into not only PD-L1 expression as a whole, but we can now look at PD-L1 expression on immune-evading tumor cells and differentiate those from PD-L1 expression on immunosuppressive macrophages, or immune-suppressive macrophages. Additionally, the presence of cytotoxic cells within the tumor compartment versus outside the tumor can be easily quantified. And then, of course, using these phenotypes together on a single section of tissue, a much greater spatial insight into distances between tumor cells and specific immune cell types within tissue section are enabled. So with this paper, it was really nice to see not only we're able to get that concordance from chromogenic DAB to the ISP multiplex immunofluorescence assay, but we're also able to get all of this complex phenotyping as well as spatial information. However, we can take this information one step further with the ability to perform a same slide H&E, and we're even able to co-register this information and stack it to generate a single image with both the immunofluorescence data as well as the H&E. So by incorporating the same slide multiplex immunofluorescence with an H&E, we are able to acquire even more information. So with the multiplex technology, there can be up to 12 biomarkers that can be combined with whole slide imaging that allows for clear single cell resolution. And these high-plex stains show you detailed phenotypic profiles and the localization of these cells within their microenvironment, while also giving you the classic pathologist annotations overlaid with cell morphology and tissue architecture. So this can generate an even greater amount of information. So next, we're able to get even more information on a tissue section, in doing so by incorporating the same slide multiplex immunofluorescence with an H&E. And with this, we can have the multiplex immunofluorescence technology with up to 12 biomarkers that can be combined with the whole slide imaging that allows for clear cellular resolution. These high-plex stains show you the detailed phenotypic profiles as well as the localization of these cells within their microenvironment on the same slide and image as a classic pathologist annotations. So this allows for you to have the cell morphology as well as the tissue architecture provided by the H&E. And so this can provide you with even more cellular and spatial information. So just to give you an example of what this can look like, in the next few slides, I have an example of how both the H&E as well as the multiplex immunofluorescence stain combined on the same exact tissue section can give you much more information. So here we're starting off just showing a region of interest of a non-small cell lung cancer with the H&E. And from the H&E alone, we're able to see the tissue architecture. We can see a single cell resolution where we can observe cell morphology. And just looking at this image, we can easily differentiate the carcinoma from areas of stroma. Looking at the individual cells, we can even tell lymphocytes versus tumor cells. However, the limiting factor here is that we can't further differentiate these lymphocyte populations. And just to highlight a certain couple lymphocytes in the upper right-hand corner, I have a couple shown in yellow arrows. So next, I'm going to switch over to the fluorescent InSituPlex image. Now this is the same exact slide, same exact region of interest. And in the upper right-hand corner, we can even see these 3 cells. So here we had stained with the T-cell activation kit. Oh, sorry, this is the PD-1 kit. With the PD-1 kit, we can easily differentiate these cell populations where we have CD3 in the red, CD45RO in the green, and PD-1 in yellow. So now looking at these 3 lymphocyte populations, we can easily differentiate in the green, in the red with the green text, we have our single positive CD3 cell. So this is our non-exhausted T cell. Below that, we have an NK cell. And then we have our exhausted T cell that's positive for both CD3 as well as PD-1. Now we're going to overlay these 2 technologies. So here we have the H&E with the 3 different lymphocyte cells that we couldn't differentiate. And then here we have the fluorescent image of all 3 combined. So we can see how we have the information where we can visualize the tumor versus stroma, the pathologist annotations, all while having our fluorescent data. So this allows us to have more of a hyper-annotated multi-marker phenotype where we can visualize that cell morphology. So a second example of how we can incorporate H&E technology with multiplex immunofluorescence technology is here we can incorporate the H&E where we can perform the pathologist annotations, and then from there we can progress to phenotyping with the fluorescent image. So here we have the whole slide tissue in the upper left-hand corner. In the circular region, we have an annotated tissue area of tumor. And in the lower left-hand corner, we have this circled region with the phenotypes being presented. So we're going to start off looking at one marker at a time. So first, we'll begin looking at tumor. And just like what was annotated in the H&E slide that we had shown previously, here we're looking at pancytokeratin as the tumor marker. And we can see that these overlap with one another, which is not surprising, but it's good to see confirmation. Next, I'm going to add T cells. So here we've then incorporated a marker for CD3. And of course, our T cells here are shown in green. What we can see with our T cells is that the T cells have actually infiltrated into the tumor region, shown in the circle, although some have also remained within the non-tumor compartment, which is, there are two areas, especially flanking either side of the tumor region. So next, we're going to further differentiate these T cell populations. And we're going to do this by adding a marker for CD8, which is a cytotoxic marker, and here it's shown in cyan. And what we can see is that there are quite a few of these cytotoxic T cells that have infiltrated into the tumor environment. And then this is going to be next paired with a stain to identify macrophages, which will be shown in magenta, and this will be for CD68. And of course, macrophages are antigen-presenting cells. So ideally, these macrophages would be priming those cytotoxic T cells that had infiltrated into the tumor area, ideally to prime them to attack the tumor cells. However, next I'm going to add the PD-L1 stain, which is going to be shown in orange. And with PD-L1, it's a marker for immunosuppressive macrophages, as well as immune-evasive tumor. And unfortunately, what we can see here is this particular individual has a very large amount of PD-L1 being expressed. So unfortunately, those cytotoxic T cells that have made their way into the tumor likely won't be performing their cytotoxic function on the tumor itself. However, it is possible that this patient might be a good candidate for anti-PD-L1 therapies based on the amount of PD-L1 being expressed. So hopefully that is good news for this particular individual. So just looking at these 5 different stains in combination with the H&E, we can see that this gives you a much better picture of what is occurring within the tumor microenvironment. And not only can we quantify this, but we can physically see all of these interactions in the tissue. And of course, as I had mentioned here, we're just looking at 5 markers. But with technologies, you're not limited to just 5. You could use 8 or even up to 12 biomarkers, and this will provide you with, of course, even more data with an even clearer picture about the tumor microenvironment and what exactly is occurring within the tissues, so that people can have a better understanding for drug discovery or patient treatment, for example. And then, of course, with this information, can go on to perform image analysis, whether it be just general whole slide quantification, whether more detailed phenotyping is required, whether we want to compare tumor versus non-tumor, or even get more in depth into spatial interactions with different cellular phenotypes. But just to give you some examples of the data that can be acquired, with whole cell quantification, we're able to, whole slide cell quantification, we can quantify each of these single markers. With that, we can go a step further where we can look at more complicated phenotypes, where we could look at double positives or triple positives, where we can identify not just tumor cells, but immune-evading tumor cells. If we wanted to look at T cells, regulatory T cells, exhausted T cells, we could do all of that and more. And we can do this either for the whole entire tissue section, or we could potentially segment out the tumor versus the stroma utilizing a mask like with cytokeratin, where we can easily differentiate tumor from non-tumor areas, and we can compare the cell number, cell frequency, or density within these two regions. And of course, this can give us a large amount of information to compare what's in the tumor versus what's present in the stroma. So just to give you one final example of the information that can be acquired from this sort of assay, in this final publication that I've shown here, cell populations and their spatial relationships were observed in skin biopsy samples taken from individuals with lupus, which is an autoimmune disorder driven by cytotoxic T cells targeting dermal components and releasing inflammatory cytokines. And B cell populations had also been involved, but up to this point, their exact function was unclear. But in this paper, the authors did want to gain a greater understanding of the roles that B cells played, and to classify inflammatory patterns of this disease through the utilization of a custom multiplex immunofluorescence panel. So this custom panel included markers for CD20, which is a classic B cell marker, MHC2 to identify antigen-presenting cells, both CD80 and CD86, which are activation and memory markers of B cells, as well as BAFFR, which is a pro-survival marker of B cells. So here are the fluorescent data incorporated in H&E to visualize tissue architecture as well as the cell morphology. So in this cohort of individuals, there were 17 different biopsies that were stained and imaged for multiplex immunofluorescence. And in this studied cohort, around 50% of the skin samples were shown to be significantly B cell infiltrated. So this confirmed a well-known observation in autoimmune disorders where T and B cells can form clusters to mimic lymphoid structures. So using multiplex immunofluorescence, it was found that there was a subset of lesional cells that showed direct co-expression of CD20, MHC2, as well as CD80 and the 86. And this strongly indicates that B cells, especially those that were arranged within clusters, have both antigen-presenting as well as T-cell activating functions. So this particular study did show that the presence of altered lymphoid populations within certain individuals have a histological assessment in regard to B cell populations, and this may actually be beneficial to better tailored treatment decisions, and that some cases may need a more targeted therapy that's targeted more towards B cell populations. So over the course of my presentation today, it's been demonstrated that quantifying cellular populations based on number, frequency, or density alone may not be enough to fully tease out patterns that could be prognostic indicators or markers of certain disease states. So that's where really looking at other patterns potentially involving spatial biology are important to learning more about. So multiplex immunofluorescence technology allows for multiple markers to be put on a single tissue section. So not only are we able to now quantify multiple complex phenotypes, but we can then incorporate a same slide H&E that will give you the multiple phenotypes, their spatial relationships to one another, all while having the ability to have a classic pathologist annotation, as well as observing the tissue architecture, as well as cell morphology. So this allows you to have a much greater amount of information on a tissue section that can be used to gain a greater understanding of the spatial environment. So in the future, I think we still have a long way to go in understanding all of the information that is placed on a single tissue section. So I think moving forward, in order to get this to the point where it could be utilized as a diagnostic, it will require a really collaborative effort between groups and just a greater understanding of these inputs. But it's really exciting to see the technology moving forward and what's going to take place in the future. So I would like to thank you all for your time today.